If all prion diseases affect the same protein, why are the diseases different?
Great question, UPM me your dick brothers, and you're on the right track with your hypotheses. In fact, it's both how the protein misfolds and where the prints tend to target accumulate, in part at least because of how it misfolds. The protein misfolding aspect is a key part of the strain concept of prints. With some prion diseases, multiple different strains have been identified. Sometimes this may be due to different amino acid sequences of the host prion protein like Phi and CJD, as you've pointed out, but that's not always the case.
In some experimental strains of scorpion mice, RML and Me7 for example, the amino acid sequences are identical but the protein is thought to misfold into subtly different conformations. Imagine different cuts of a key, take those different cuts of a key blank and consider how specific they are for a given block.
That's thought to be what's driving differences in neuropathology and ultimately clinical symptoms as well. Different strains can have different incubation periods and different symptoms like weight loss, behavioral changes, etc. The misfolded prions preferentially target different areas of the brain and other tissues because those areas may have prions with different modifications, sugars, etc. added by the specific cell types that produce. Last comment is an area of active discussion presently.
The researcher who first determined that prion diseases involve solely misfolded proteins, Stanley Prusiner, is one of many arguing that other protein misfolding disorders like Alzheimer's and Parkinson's disease should be reclassified as prion diseases.
Although they involve completely different proteins, a beta and alpha-synuclein, respectively, the process involved may be best grouped together with the misfolding process that prints undergo along the lines of how coronaviruses, influenza viruses, and norovirus are all viruses, just different families of viruses.
With several protein misfolding disorders, there is often one primary protein that is misfolding, but there may be others as well, like tau, that can accumulate, kind of like an innocent bystander process as the cell protein folding refolding infrastructure becomes dysregulated. I can update with specific references, but Reddit is always terrible about blocking primary literature links.